already started.
Good morning. So today for the journal club topic, I will be discussing the article Dental Calculus Formation Rate, the role of salivary proteome and metaproteome. This article was published in the Journal of Clinical Periodontology in 2025 with an impact factor of 6.8. So coming to the terminologies that are discussed in this journal for discussion in general. The first one is the salivary proteome. It is a complete set of the human-derived proteins present in the saliva at a given particular time, reflecting the biological functions such as the immunity and the mineral balance and the antimicrobial defense actions. Also, the metaproteome is the entire collection of microbial proteins that are found in a sample. In this case, they have taken the salivary sample and it represented the active functions of the oral microbiota. Dental calculus formation rate, a quantitative measure of how quickly the mineralized dental plaque is forming on the teeth over time, influenced by individual saliva composition, microbial activity and oral hygiene. The acquired pellicle formation, we already know it is a protein-rich thin film that forms on the surface of teeth seconds after brushing, derived from salivary proteins, which can act as a barrier for the base for the plaque and the calculus formation. And lastly, the enzyme enrichment analysis. They did the statistical method to identify which enzymes or pathways are overexpressed in a data set, like the salivary proteins that they quantified in this journal, offering insights into the dominant biochemical activities. So coming to the novelty, aims and objectives, what was the novelty of the study? It was the first study to comprehensively integrate the salivary proteomics and the microbial metaproteomics to explore individual variation in dental calculus formation rates, identifies the specific host and the microbial proteins, specifically the calcium binding proteins and the urease enzymes that are expressed in the saliva associated with rapid calculus development, and moves beyond traditional plaque assessment techniques by combining high resolution mass spec with bioinformatics to decode the biological signatures that will lead to formation of the calculus in the study population. So coming to the aims of the study, to investigate whether the differences in the quantification of the salivary proteins and the bacterial enzyme activities explain why some people develop calculus faster than others, to understand the biochemical environment of saliva in fast versus slow calculus forms. So the objectives of the study was to categorize individuals into fast, moderate and the slow calculus formers using standardized plaque indexing that they used, perform mass spec and the bioinformatic assays of saliva samples to quantify both human and the microbial proteins, and compare the functional pathways such as the urease activity in general and the calcium phosphate binding capacity across groups, and identified lastly the protein biomarkers and enriched enzyme classes that may predict calculus susceptibility. So coming to the materials and methodology, this study was centered in Canada. So saliva samples were collected from 26 healthy participants categorized into slow, moderate and fast dental calculus formers based on the clinical indices. Unstimulated whole saliva was collected for the study under standardized conditions and stored under -80°C. The use of LCMS to analyze the salivary proteome and the metaproteomes were done, and the statistical comparisons were made between the calculus formation rates on the three groups to detect significant proteomic differences of the levels of proteins related to host defense mineralization strategies and microbial metabolism. So coming to the statistical analysis, they did multiple statistical analysis tests such as ANUVA, TUPE's post hoc test, false discovery rate correction test and principal component analysis and enrichment analysis. These were done to compare for the protein abundance, pairwise comparisons, and to do visualize clustering of the proteomic profiles, and use the gene ontology using bioinformatics, and to have the information on the KEC pathway to identify overexpressed biological process and the enzymes helpful in the formation of the calculus over the three groups. The parameters that were compared were salivary proteins that were host derived, that were the levels of the calcium binding proteins, mucins, carbonic anhydrase 6 and proline rich proteins, microbial proteins quantifications and enzyme level quantifications such as urease, glycosidase and proteins involved in the biofilm formation better, and mineral ion transport was also recognized and watched out for, and functional pathways and enzyme classes including mineralization strategies, pH regulation, proteolysis, and ammonia production was done. So coming to the outcomes, from the groups of the fast calculus formers and the slow calculus formers, the fast calculus formers had significantly high levels of host proteins that were rich in calcium, specifically the SA100AA and A9, and carbonic anhydrase 6 levels. Microbial proteins, urease and other ammonia generating enzymes were present. PCA analysis showed that the distinct separation between the fast and the slow formers were there, confirming biological differences in the salivary environment. Enrichment analysis pathway revealed that the alkaline pH modulating and mineralization proteins were overexpressed in the fast group as compared to the slow group, linking them to the accelerated calculus formation. So coming to the results, out of the 26 participants, 3 were males and rest were females. 23 were females. So in that, 11 were rapid calculus formers, 15 were slow calculus formers with no baseline health or demographic differences. When we come about the salivary chart, this is the significant result of the study, that is the slow formers had a significantly more negative zeta potential than rapid formers, which may help keep calcium dissolved and